welcome to the first of our Biology 121 online videos. Today we're talking about photosynthesis. We're going to go through what are called the light-dependent reactions in photosynthesis. We're going to talk about how photons get absorbed by plant cells to cause excited electrons that move through an electron transport chain, producing NADPH and ATP, which will later be used to form sugars. But we'll get back to the sugars in our next video on Friday. So let's first reiterate that photosynthesis is the opposite of cellular respiration. We were talking about cellular respiration just before all of this break happened. And so I just want to reiterate that while mitochondria are a formerly independent form of bacterial life that was absorbed by eukaryotic cells a very long time ago, chloroplasts perform the chloroplasts that perform photosynthesis are very, very similar to mitochondria, except that where the mitochondria take sugars and break them down to form ATP, chloroplasts take the energy from sunlight and use it to form sugars. So again, we have a formerly symbiotic bacteria performing cellular functions that are really important for capturing energy so that organisms have that energy available to be used. So a little recap of cellular respiration. We talked about it as a four-step process, starting with glycolysis, where sugar molecules are broken down into smaller, what are called pyruvates, producing a little bit of ATP as they do so. And then in the second step, that pyruvate is processed into a chemical called acetyl-CoA, which is then enters into what's called the Krebs cycle, where that acetyl-CoA gets further broken down and broken down. In the Krebs cycle, you get a little bit of ATP production. You also form a bunch of molecules of NADH and FADH2. And what those are, again, are kind of, we've referred to them as almost like battery-like storage molecules that use energized electrons attached to these larger complexes to carry the energy in those electrons. And then that electron transport chain is where that energy gets transported from one molecule to another until eventually ATP synthase receives that energy and can use it to charge up some ATP molecules from ADP molecules. So photosynthesis is exactly the opposite of this, in that we're still going to see a cyclical process in which sugar is involved, but now instead of the sugar being broken down in that cycle, we're going to be forming that sugar. But before we get to that, that's step 4. Before we get to that, we have to absorb the light. That's step 1. We have to transport electrons that are highly energized by that light from the low energy NADP plus is going to become a high energy NADPH, carrying that high energy electron with it. We're going to form a little bit of ATP, and then, and we'll talk about this one on Friday, that carbon is going to get fixed, and the Calvin cycle is going to form a sugar using the energy from that ATP. Let's start with a little bit of an introduction to the structure of a chloroplast. Now, the chloroplast is a organelle inside plant cells that where all of the photosynthesis happens. If we start sort of on the very far left where it says chloroplast envelope, you see it has an outer membrane, an intermembrane space, and then an inner membrane. So it's got a double membrane. It's have two separate membranes. And again, this is because chloroplasts used to be, back in early evolutionary history, chloroplasts were a separate photosynthetic bacterium that got absorbed by another cell, a eukaryotic cell, that then became the first plant. So we have, again, just like with mitochondria being absorbed, and then because they were very, very functional, the chloroplast was absorbed by another cell, and then that cell said, hey, if I just let this bacterium keep doing what it's doing instead of eating it, I get all this sugar out of it. Great deal. So that double membrane, the outer membrane, is actually provided by the plant cell, and the inner membrane is provided by the chloroplast. That's why we have this double membrane system. The thylakoid, so working further around there, I'm actually going to switch diagrams here because I think the second diagram is a little easier to read. So again, if we go around this kind of clockwise, starting from the upper left, The name of the organelle is a chloroplast, the outer membrane we just talked about. If we go further to the right, we see the granum. Now, a granum is a stack of discs, that light green colored stack there that looks like a giant stack of pancakes. That's a granum. When we've got all of these discs together in a pile like that, we call that pile up a granum. The next word in the sequence is going around clockwise is lumen. The lumen's pointing towards this dark green space that's inside those individual discs. The lumen is the fluid that's inside those discs. Now the next word around, as we go around the circle here, is thylakoid. Thylakoid, that's a tough one, right? Everyone say thylakoid. It's an old Greek word. I don't know what it's supposed to mean, but it refers to the individual discs that form up those granums. So if a granum is a stack of pancakes, the thylakoid is an individual disc-like pancake itself. The stroma is the next word there. So we said the lumen is the liquid that's inside a thylakoid. The stroma is the liquid outside a thylakoid. There's an important distinction because they actually have different chemicals in those two different liquids, and that's going to be actually really important a little bit later. And then the last word as we go around the circle there is the inner membrane. So that inner membrane, again, is just the one provided by the chloroplast itself. So I'm going to go back to the other picture for a second. So we can see here we have the thylakoid has a thylakoid space, and the space inside of it is full of lumen. And then there's a thylakoid membrane. That's what thylakoids are made of. They're actually a membranous disc. And that's really important because that membrane is going to turn out to be where a lot of the work actually happens. Just like with mitochondria, it's in the mitochondrial membrane that all of these proteins that were involved in the enzyme functions of the Krebs cycle, the Krebs cycle were involved. No, citric acid, that cycle was involved in the membrane itself. We're going to see this similar thing happening here where the thylakoid membrane is carrying proteins in it embedded in that membrane. We'll get to that in a moment. So let's talk about this key vocabulary. I'm going to go down column by column. So in the first column, the first word is chloroplast. The chloroplast is the organelle itself where photosynthesis happens. It's composed of all of these other subpieces. A granum is a stack of thin discs made out of membrane. Each disc is called a thylakoid. So that's our first column. The second column, the lumen is the liquid inside the thylakoid, and the stroma is the liquid outside the thylakoid but still inside the chloroplast. The third column, we have photons. Now, a photon is the smallest single particle of light. Light is weird. Light is a kind of electromagnetic radiation, and what's interesting about that is that it means that it is actually both simultaneously a particle, a piece of matter, and a wave, an insubstantial waveform. We've talked in class previously about how different wavelengths of light have different colors, and also how things like X-rays and gamma rays and microwave and radio are all different wavelengths of light, of electromagnetic radiation. So we're going to be talking when we talk about these photons, we're only really talking about the photons that are in the visible spectrum of wavelengths of light. Chlorophyll is a chemical that specifically is a kind of pigment, there's our last word, and any pigment is just a chemical that absorbs certain frequencies of light. So chlorophyll is a pigment that absorbs all of the pigments of light except for green light. So the green light reflects off, and that's why plants look green. So let's move on. We start when we're absorbing light with what's called an antenna complex. And it's called an antenna because all an antenna is anything that absorbs electromagnetic radiation. The antenna for your radio in your car can receive radio waves. Radio waves are a form of electromagnetic radiation. They are fundamentally not different from visible light. The antenna in your cell phone can absorb and also produce microwave radiation. So we call the first step of photosynthesis the antenna complex because that antenna is the chlorophyll itself absorbing electromagnetic radiation in the form of visible light. So let's go through this. Solar energy, specifically a photon, a single photon, gets absorbed by a network of molecules called a photosystem. The mix of molecules is different from one species to another. Typically, chlorophyll is a key molecule. There are other pigments involved. There are actually two different kinds of chlorophyll. Chlorophyll A is the most common, but there are some plants that have chlorophyll B in different amounts as well. There's also other pigments called carotenoids that are more yellow and orange and red. So if you think about like plants sometimes have very red leaves, like the Japanese sugar maple is never green leafed. It's always bright red leaves. There are certain kinds of cabbage and other kinds of brassicas that have red leaves, like a red cabbage, right, or a radicchio. Those red-colored leaves don't have very much chlorophyll. They're depending on other pigments to absorb sunlight. But chlorophyll is by far the most common, and that's why most plants have green leaves that absorb sunlight, bouncing off the green light only. And then that sunlight, the photons from that sunlight, elevates the charge on an electron. So that electron becomes more energetic than the other electrons around it. And the energy in that electron gets passed around among many different chlorophyll molecules, almost like a game of hot potato. Like the chlorophyll molecules are like, no, you take the electron, no, you take the electron, no, you take the electron, right? So let's take a look at what that looks like. So here we have a photosystem antenna complex, and if we go around this again, sort of starting in the upper left corner and working our way around a little bit, we see the photon is represented as this zigzagging yellow line. That's really common. You're going to see that a lot as an indication of energy coming in, specifically solar energy coming into the plant. It strikes a chlorophyll molecule, and that molecule then absorbs that solar energy from the photon, excites an electron. So we've got one highly charged electron, and that electron is going to bounce around. We see the yellow line bouncing from one molecule of chlorophyll to another until eventually it gets to the edge of the photosystem, where a chlorophyll molecule that is in what's called the reaction center, which just means that it's out at the edge of the photosystem where it's accessible to the rest of the cell, or to the rest of the thylakoid. That electron then gets shot out and it leaves the chlorophyll entirely. Let's take a look at it in this picture. So again, on the right-hand side of your screen, you see the light coming in. This time it's drawn in pink instead of yellow, but that's fine. And we see the electron bouncing through in this kind of diagonal line going downwards until it reaches this reaction center chlorophyll down at the edge and kind of outside, disconnected from the antenna. And then that blue line that's zigzagging across with a little circle with an E in it represents the electron being shot out and ejected from the chlorophyll. And it shoots its way through this reaction center until it reaches an electron acceptor, which is a molecule that is ready to take on an electron. Remember that NADPH, right? That's going to be our primary electron acceptor in photosynthesis. So that electron, that highly charged electron, now has left the antenna and is ready to go do some work out in the rest of the photosystem. So now we're going to get into these steps 2 and 3, because now that we have an electron that is charged up and has left the antenna, we're ready to transport that electron until it gets used to produce energetic full batteries, as we've talked about before. So the energy harvested by the photosystems are transferred through NADPH, that's the electron carrier, and used to produce ATP. This is a multi-step process. There's several enzyme complexes. I was watching a video earlier today that said there's at least 99 different enzymes involved in this process. You don't need to learn all 99 names. Don't worry about it. So this happens inside of a chloroplast in the thylakoids, and specifically these chemicals, these 99 different enzymes, are embedded in the membranes of the thylakoid. So let's take a look at this pictorially. Once again, as you're used to seeing, whenever you see these little like circles or ovals with little tails, squiggly tails that are pointing towards each other, you know we're looking at a membrane. So this is the membrane of the thylakoid. The top of this membrane represents the stroma, the part of the chloroplast that is outside the thylakoids, and the bottom of it represents the inside of the thylakoid. So that's the lumen. So what we see in 3 bands, we have a blue band that is the stroma, we have a kind of yellow band that is bordered in gray that is the membrane itself, and then we have the tan band at the bottom represents the lumen. So again, we see light shown as this kind of zigzagging line hitting this photosystem. Those light green circles this time represent the chlorophyll. The electron energy gets passed from chlorophyll to chlorophyll through that photosystem 2, and then eventually becomes an electron that is ejected through the center in this drawing, and it goes out, and that orange arc represents the electron actually leaving the photosystem and entering the electron transport chain. Now, a brief moment. Why is photosystem 2 here before photosystem 1? The answer is because people figured out about photosystem 1 before they learned that there was a photosystem 2. So we as scientists discovered this photosystem that charges up ATP through NADP reductase before we learned about the photosystem that leads into this electron transport chain. And so for that reason, the one that happens first is called photosystem 2, and the one that happens second is called photosystem 1, because we learned about them and we discovered them in the opposite order. So that electron transport chain is sometimes also called the B6F complex. That's going to show up in the next picture, so make a note of that. That produces ATP, and it uses energy from electrons that are stripped from protons. So we'll come back around to that in a moment. Photosystem 2 also absorbs light. And again, exactly like in photosystem 1 also absorbs light. The second photosystem, photosystem 1, also absorbs light, and the electrons bounce through those chlorophylls until an electron is ejected, so they work exactly the same way. And this time, a molecule called NADP reductase, which is in the oxygen redox reaction system, is going to accept that electron. It's going to absorb that electron, and then that reductase is going to give the electron off, or give the energy from the electron off to that ATP synthase. Now, ATP synthase is an enzyme that does exactly what it says. It synthesizes ATP. It makes ATP using energy related to the buildup of protons inside the membrane through an osmotic pressure. So let's take a look at this from another perspective. This is a much more abstract drawing, but it's exactly the same thing. So you see this 2-color green PS2, 2-color green PS1 further to the right. Those were the photosystem 2 and photosystem 1 that you saw in green just a moment ago. At the top of the screen, we have the stroma, then there's a tan band representing the thylakoid membrane, and then the lumen on the inside. So it's structured exactly the same as the last drawing. It's just a little more abstracted. The electron transport chain here is shown in blue and says CYT B6F. Remember I said B6F is going to show back up? B6F in the middle there, that electron transport chain, aka B6F, B6F. So what's happening here is photosystem 2 is using some of the energy from that electron. Well, it's actually stripping off of the hydrogen in this H2O. It's stripping the electron off of that, making an H plus proton. So a hydrogen ion is called a proton inside the thylakoid. And that electron then gets passed off and is used to pull another proton, another H plus molecule, inside the thylakoid. So now what we're getting is this buildup of H pluses inside the thylakoid. Photosystem 1, the second one, see, I'm making a mistake. Photosystem 1, the second photosystem in the sequence, absorbs more light and charges up an NADPH. So it's again drawing electrons out and using them to store that electron in NADPH. That NADPH is out in the stroma. So now we have this highly energized particle that's just sitting outside the thylakoid, but still inside the chloroplast, ready to have its energy used. And then finally, ATP synthase uses the fact that there's a buildup of pressure of hydrogen ions, H pluses, inside the thylakoid, and by releasing that pressure, it's able to do some work. So when it lets that pressure out, it takes the energy of that pressure release and uses that energy to charge up an ATP molecule using an ADP in the way. Remember, ADP is the low energy version, ATP is the high energy version, so it's charging up energy into that ATP down at the end there where we see the ADP-ATP swoop. Finally, let's just summarize real quick. What we just saw in both of those two drawings was the second and third step, the electron transport chain and the ATP formation that happened in those two separate photosystems. So photosystem 2 enables the electron transport chain, photosystem 1 enables the ATP formation, and this two-step process is very similar to how the electron transport chain in respiration forms ATP, but here the electrons are charged up by light rather than electrons being charged up by the digestion of sugar or other chemicals. So, right now, where we've gotten to is the light-dependent reactions. Again, both parts of this photosystem, these two photosystems, both photosystems require light in order to function. So, what we've got thus far is ATP and NADPH, these two different charged-up batteries, ready to be used. The next step, step 4, is going to be what's called the light-independent reactions, because now that we have light energy stored in the form of chemical energy, we can convert that chemical energy to a different kind of chemical energy without needing any light to do it. That's going to be Friday's video, so I'll talk to you then.